So here you are, lying in the dark, and your body is doing something right now that you're completely unaware of. Somewhere inside you, at this exact moment, a white blood cell is crawling along the wall of a tiny blood vessel, squeezing itself between the cells that line that vessel wall, and pushing out into the surrounding tissue because it detected a faint chemical signal, a whisper really, that something nearby isn't quite right. Maybe a bacterium slipped through a microabrasion on your skin that you don't even remember getting. Maybe a virus hitched a ride on something you touched 6 hours ago. It doesn't matter. The point is, while you're settling in and your conscious mind is winding down, there's an entire civilization of cells working a night shift inside you, and they have been every single night of your life and every single day too, and they are spectacularly good at what they do. Let's talk about that. Let's talk about your immune system. The whole thing, from the ground up, slowly, because it's one of the most fascinating systems that exists anywhere in nature, and it deserves a proper telling. Not a rushed overview, not a diagram you'd see in a textbook. Just the story. How we figured it out, how it actually works, why it fails sometimes, and why it succeeds so often that you barely notice it's there. Because that's the real marvel, isn't it? The immune system's greatest trick is that when it's working well, you feel nothing at all. You just go about your day, completely unaware that a small war was fought and won somewhere between your elbow and your shoulder while you were eating lunch. But here's the thing. Humans didn't always know any of this. For most of recorded history, disease was a mystery, an absolute, terrifying mystery. People got sick and nobody understood why. The explanations ranged from bad air to angry gods to alignment of the planets. And honestly, if you were living in, say, ancient Athens around 430 BC, you'd have no reason to think otherwise. That's where our story starts, actually, because something happened in Athens during the Peloponnesian War that planted one of the earliest seeds of immunological thinking, even though nobody at the time had the faintest idea what an immune system was. Athens was under siege. Sparta and its allies had pushed the Athenians back behind their city walls, and the population was packed in tight. Way too many people in way too small a space. And then a plague hit. We still argue about exactly what disease it was. Typhus, maybe, or typhoid fever, possibly smallpox. The descriptions that survive are brutal. The historian Thucydides, who caught the disease himself and survived, wrote about it in detail. Fever, redness of the eyes, bleeding in the throat, vomiting, skin covered in ulcers. People died by the thousands. Maybe a quarter of the Athenian population was wiped out over about 4 years. But here's what Thucydides noticed. And this is the part that matters for us. He wrote that the people who had recovered from the plague could safely nurse the sick without catching it again. He didn't know why. He couldn't explain the mechanism. He just observed it. People who survived seemed to be protected afterward. That's immunological memory. That's your adaptive immune system doing its thing, building a record of what it's seen so it can respond faster next time. Thucydides described it 2,400 years before anyone understood a single molecule involved in the process. He just watched, and he wrote down what he saw, and that observation sat there in the historical record like a seed waiting for soil. Now, you might think that observation would have kicked off a flurry of investigation. It didn't. For roughly 2,000 years after Thucydides, the dominant theory of disease in the Western world was the miasma theory. The idea that illness came from bad air, from foul-smelling vapors rising out of rotting organic matter, swamps, and waste. And look, it wasn't a crazy theory given what people could observe. Places that smelled terrible, open sewers, swamps, battlefields after a fight. Those were places where people got sick. The correlation was real, even if the causation was completely wrong. The air wasn't making people sick. The mosquitoes breeding in the swamp water were. The bacteria multiplying in the sewage were. But nobody could see bacteria. Nobody could see viruses. You needed a microscope for that, and microscopes didn't exist yet. Meanwhile, though, on the other side of the world, something remarkable was happening. In China, possibly as early as the 10th century, the exact dates are debated, and some scholars push it later, to around the 15th or 16th century, practitioners had developed a technique for preventing smallpox. Smallpox was one of the great killers of human history. It's hard to overstate how terrifying this disease was. It killed roughly 3 out of every 10 people who caught it, and those who survived were often left with deep, disfiguring scars. Entire populations that had never been exposed, indigenous peoples in the Americas, for instance, were devastated when European colonizers brought it across the ocean. But Chinese practitioners noticed something similar to what Thucydides had noticed. Survivors didn't get it again, and they took that observation and did something incredibly bold with it. They took dried smallpox scabs, actual material from the pustules of people who'd had mild cases, and they ground it into a powder, and they blew it into the nostrils of healthy people. Sometimes they'd take a bit of pus from a smallpox sore and scratch it into the skin instead. The idea was to give the person a mild case of the disease so their body would learn to fight it off. Think about what that means. Think about the courage, or maybe the desperation, that required. You're deliberately exposing a healthy person to one of the deadliest diseases on Earth, betting that the exposure will be mild enough to survive, but strong enough to protect. And it worked. Not perfectly. Sometimes the person got a full-blown case and died. The fatality rate from this procedure, which came to be called variolation, was somewhere around 1 to 2%. That sounds scary, and it was. But compare it to the 30% fatality rate of naturally acquired smallpox, and suddenly a 2% risk looks like a very rational bet. This practice spread along trade routes. It reached the Ottoman Empire, where it was widely practiced by the early 18th century. And this is where one of those wonderful human stories enters the picture. A story about a woman whose curiosity and stubbornness changed the course of Western medicine. Her name was Lady Mary Wortley Montagu, and she was the wife of the British ambassador to the Ottoman Empire. She arrived in Constantinople in 1717, and she was already intimately familiar with smallpox. She'd had it herself in 1715, and it had left her famously beautiful face scarred. Her brother had died of it. So when she learned that the Ottoman women had this practice of deliberately inoculating people against the disease, they called it engrafting. She was immediately fascinated. She watched the procedure. She asked questions, and then she did something that took real nerve. She had her own son inoculated right there in Constantinople, with the embassy surgeon watching. When she returned to England, she became a fierce advocate for the practice. And here's where you see how science and society collide, because the resistance she faced was enormous. The medical establishment pushed back. The church pushed back. People were horrified by the idea of deliberately giving someone a disease. There were class prejudices at work too. This was a folk practice from the East, promoted by a woman in an era when women's medical opinions were routinely dismissed. Lady Mary fought for it anyway. She arranged a public demonstration where condemned prisoners at Newgate Prison were inoculated. They survived and didn't catch smallpox when later exposed. She had her daughter inoculated publicly in London. Slowly, very slowly, the practice gained ground among the British aristocracy. Even members of the royal family were eventually inoculated, but variolation was always a gamble. That 1 to 2% death rate was real. People did die from the procedure, and the inoculated person could spread full-blown smallpox to others during their mild infection. It was a tool, but it was a dangerous, imperfect tool. What the world needed was something safer. And that brings us, of course, to Edward Jenner. You've probably heard his name. He's often called the father of vaccination, and the story is usually told as a neat little fairy tale. Country doctor notices milkmaids don't get smallpox, tests cowpox as a vaccine, saves the world. The real story is messier and more interesting than that. Jenner was a country physician in Gloucestershire, England, in the late 18th century. He'd been inoculated against smallpox himself as a boy, and the experience had been miserable. Weeks of illness, bleeding, purging, the whole brutal protocol that accompanied variolation at the time. So he had personal motivation to find something better. And yes, there was a folk belief in the English countryside that milkmaids who caught cowpox, a much milder disease that caused sores on the hands, seemed to be protected against smallpox. Jenner didn't invent this observation. It was common knowledge among farming communities, but he was the one who decided to actually test it systematically. On May 14, 1796, Jenner took material from a cowpox sore on the hand of a milkmaid named Sarah Nelmas and scratched it into the arm of an 8-year-old boy named James Phipps. The boy developed a mild fever and some discomfort, but recovered quickly. Then, about 6 weeks later, Jenner did something that would get you arrested today. He deliberately exposed young James to actual smallpox. He variolated the boy with fresh smallpox material, and James didn't get sick. He was immune. Now, let's be honest about the ethics here for a second. Jenner deliberately exposed a child to a lethal disease to test a hypothesis. The boy couldn't meaningfully consent. He was 8 years old, and he was the son of Jenner's gardener, which introduces an obvious power dynamic. By modern standards, this experiment would be unthinkable. But in the context of the 18th century, when smallpox was killing hundreds of thousands of people across Europe every year, Jenner's gamble was seen differently. And it worked. It worked spectacularly. Jenner published his findings in 1798. He called the procedure vaccination, from vacca, the Latin word for cow. And the reaction was, well, it was mixed. Some people embraced it immediately. Others were skeptical. There were satirical cartoons showing people growing cow parts out of their bodies after being vaccinated. The medical establishment was divided. But the evidence kept accumulating. Vaccination was safer than variolation, much safer. And it worked. What Jenner couldn't tell you, what nobody could tell you in 1798, was why it worked. He had no idea what a virus was. He didn't know about antibodies. He didn't know about T cells or B cells or any of the molecular machinery that makes immunity possible. He just knew that exposing someone to cowpox protected them from smallpox. The mechanism was a complete black box. And that's actually a theme that runs through the entire history of immunology. The practice often came first, and the understanding came later, sometimes much later. People figured out what worked by observation, by trial and error, by desperate experimentation during epidemics, and then spent decades or centuries figuring out why it worked. Your immune system was protecting you for hundreds of thousands of years before any human being had the slightest clue it existed. The cells were there. The antibodies were there. The whole intricate system was humming along perfectly well without anyone understanding it. So the question that haunted the 19th century was this: what actually causes disease? Because until you answer that, you can't really understand immunity. You can't understand what the body is defending against if you don't know what the attacker is. And answering that question required a revolution in thinking, and a microscope, and a very stubborn French chemist who wasn't even a doctor, and a fierce German rivalry that would reshape medicine forever. The chemist's name was Louis Pasteur, and the German was Robert Koch, and the story of how they circled each other, sometimes cooperating, often competing, occasionally being petty in ways that are honestly kind of delightful, is one of the great dramas in the history of science. But before we get to their rivalry, we need to talk about what Pasteur was doing in the 1850s, because it started with something you might not expect. It started with wine. Specifically, it started with wine going bad, and a group of frustrated French winemakers who wanted to know why their product kept turning sour, and a young chemist who agreed to look into it and ended up changing the entire course of medicine. See, Pasteur wasn't trained as a biologist. He was a crystallographer, a chemist, someone who spent his early career studying the shapes of tiny crystals under polarized light. And that background turned out to matter enormously, because it meant he approached biological problems with a chemist's rigor, with controlled experiments, with careful measurement, with an obsessive attention to what was actually happening at the molecular level. So, when these winemakers in Lille came to him in the mid-1850s and said, "Look, we've got a problem. Our beet juice fermentations keep going wrong. The alcohol is turning to vinegar. We're losing money. Can you figure out what's happening?" Pasteur didn't just shrug and say it was some vague chemical reaction. He got his microscope out, and what he saw changed everything. He saw tiny living organisms in the spoiled wine, little rod-shaped creatures, different from the round yeast cells you'd find in healthy fermentation. And he realized, and this was genuinely radical at the time, that fermentation itself wasn't just a chemical process. It was a biological one. Living microorganisms were doing the work. The yeast were converting sugar to alcohol, and these other organisms, these bacteria, were converting the alcohol to acetic acid, which is vinegar. That's why the wine was going sour. It wasn't some mysterious chemical decay. It was an infection, a contamination by unwanted microbes. Now think about what that implies. If tiny invisible organisms can spoil wine, can they spoil other things? Can they spoil you? Can they get inside a human body and cause disease? That idea, what we now call germ theory, wasn't entirely new. People had speculated about it for centuries. A physician named Girolamo Fracastoro had proposed something like it back in 1546, suggesting that disease might spread through tiny seed-like entities, but nobody had the tools or the evidence to prove it. Pasteur's wine work didn't prove it either, not directly, but it cracked open the door. It demonstrated that microorganisms could cause specific chemical changes in their environment, that different organisms cause different effects, and that you could study this systematically. And Pasteur kept pushing. In the early 1860s, he tackled one of the biggest scientific debates of his era: spontaneous generation. A lot of scientists still believed that life could arise spontaneously from non-living matter. You leave meat out, maggots appear. You leave broth in a flask, it goes cloudy with microbes. Where did they come from? Many people said they just emerged. Pasteur said no. He said they came from the air, from contamination, from organisms that were already out there, invisible and everywhere. And he designed these beautiful experiments to prove it. His famous swan neck flask experiments, where he boiled broth to sterilize it and then drew out the neck of the flask into a long S-shaped curve. Air could still get in, but any dust or microbes would settle in the curves and never reach the broth. And the broth stayed clear for days, weeks, months. But if you broke the neck off and let air flow straight in, the broth went cloudy within hours. Life wasn't appearing from nothing. It was drifting in from everywhere. This was a huge deal. It meant the world was full of invisible organisms, floating in the air, landing on surfaces, getting into things. And once you accept that, the next logical step is terrifying and obvious. They're getting into people too. They're getting into wounds. They're getting into lungs and guts and blood. And maybe, just maybe, that's what disease actually is. Pasteur wasn't the only one thinking this way. Over in Germany, a quiet, methodical country doctor named Robert Koch was approaching the same problem from a completely different angle. Koch was everything Pasteur wasn't, reserved where Pasteur was theatrical, meticulous where Pasteur was intuitive, German where Pasteur was defiantly, passionately French. And that last part mattered more than you might think, because the Franco-Prussian War of 1870 had left deep wounds between the two countries. France had been humiliated. Pasteur took it personally. He reportedly returned an honorary degree from a German university in protest. So when Koch started making breakthroughs that rivaled and sometimes surpassed Pasteur's own work, the competition became not just scientific but national, personal, and occasionally bitter. But here's the thing about Koch. His contributions were extraordinary. In 1876, he did something nobody had ever done before. He took an animal disease, anthrax, and really proved, with airtight experimental logic, that it was caused by a specific bacterium, not just associated with it, not just found near it, but actually caused by it. He isolated the bacterium from sick animals, grew it in pure culture outside the body, injected it into healthy animals, watched them get sick with the same disease, and then isolated the same bacterium from those newly sick animals. That closed the loop completely. It was cause and effect, demonstrated beyond any reasonable doubt. This methodology became formalized as Koch's postulates, a set of criteria for proving that a specific microorganism causes a specific disease, and those postulates became the gold standard for decades. They're still taught in every microbiology course today, even though we now know they have limitations. Some pathogens can't be grown in pure culture. Some cause disease only in certain hosts. Viruses broke the rules entirely because they can't reproduce outside living cells. But in the 1870s and 1880s, Koch's postulates were revolutionary. They gave scientists a rigorous framework for linking specific germs to specific diseases, and that framework unleashed an absolute flood of discoveries. Koch himself identified the bacterium that causes tuberculosis in 1882, Mycobacterium tuberculosis, and the one that causes cholera in 1883. These were two of the most devastating diseases in human history, and suddenly they had names. They had faces under the microscope. They had specific organisms you could point to and say, that's the enemy. Think about what that meant for the immune system story. For the first time, scientists knew what the body was actually fighting. Not miasmas, not bad humors, not divine punishment. Specific, identifiable, living organisms, bacteria. And once you know the enemy, you can start asking much more precise questions about the defense. Meanwhile, Pasteur wasn't sitting idle. He was racing Koch at every turn, and in the late 1870s and 1880s, he made his own monumental contributions, not to identifying pathogens, but to fighting them. And this brings us back to vaccination, because Pasteur is the one who connected Jenner's old cowpox trick to a general principle. The story goes, and there's some debate about the details, but the broad strokes are well documented, that Pasteur was working with chicken cholera in 1879. He had cultures of the bacterium that caused the disease, and he'd been injecting them into chickens, which would reliably get sick and die. But then something happened. His assistant, Charles Chamberlain, went on vacation and left some cultures sitting on the bench. When they came back and injected chickens with these old, stale cultures, the chickens got mildly ill but recovered. And then, here's the key moment. When Pasteur injected those same chickens with fresh, virulent cultures that would normally be lethal, the chickens survived. They were immune. Pasteur grasped immediately what had happened. The old cultures had weakened, attenuated to use the technical term. They'd lost their ability to cause severe disease, but retained their ability to trigger the body's defenses. It was Jenner's cowpox principle, but generalized. You didn't need to find a naturally mild relative of the disease. You could take the actual pathogen and weaken it deliberately, and then use that weakened version as a vaccine. Pasteur reportedly said, with characteristic flair, that he was honoring Jenner by extending the word vaccination to cover all such procedures, not just smallpox. He went on to develop vaccines for anthrax and rabies. The anthrax vaccine was demonstrated in a famous public trial at Pouilly-le-Fort in 1881, where vaccinated sheep survived a lethal injection while unvaccinated ones died, all in front of skeptical onlookers and the press. It was dramatic, it was convincing, and it made Pasteur a national hero. The rabies vaccine, developed in 1885, was even more dramatic. Rabies was essentially a death sentence once symptoms appeared, and Pasteur used his vaccine to save a young boy named Joseph Meister, who'd been badly bitten by a rabid dog. The treatment worked. The boy survived, and Pasteur became, arguably, the most famous scientist in the world. But here's what's fascinating from our perspective, the immune system perspective. Pasteur knew his vaccines worked. He could demonstrate it publicly, spectacularly, undeniably. But he still didn't really know how they worked. What was happening inside those chickens, those sheep, that little boy? What was the body doing with the weakened pathogen that prepared it to fight the real thing? Pasteur had theories. He thought maybe the attenuated organisms used up some nutrient the pathogen needed, starving it out. He was wrong about that, as it turned out. The real answer was far more interesting and far more complex, and it would take an entirely different kind of scientist to start uncovering it. Because while Pasteur and Koch were battling over who deserved credit for germ theory, while they were identifying pathogens and developing vaccines, a quieter revolution was beginning in laboratories across Europe. Scientists were starting to look not at the attackers, but at the defenders. They were starting to ask, what does the body actually do when a pathogen gets in? And the answers they found would split the scientific world into two warring camps for decades, a debate so fierce, so personal, and so consequential that it makes the Pasteur-Koch rivalry look like a friendly disagreement over dinner. On one side was a Russian zoologist working in Italy, who looked through his microscope one morning and saw something that made him leap from his chair with excitement. His name was Elie Metchnikoff, and what he saw was a tiny transparent starfish larva, and inside it wandering cells that were doing something no one had properly noticed before. They were eating things. Specifically, they were surrounding foreign particles, tiny splinters, bits of debris, and engulfing them, pulling them inside themselves, and digesting them. And Metchnikoff, watching this happen in real time under his microscope in Messina, Italy, around 1882, realized he was looking at something fundamental, not a chemical reaction, not some passive barrier, an active cellular defense, living cells roaming through the body of this tiny sea creature, hunting for things that didn't belong and devouring them whole. Now, to appreciate why this was such a big deal, you have to understand where Metchnikoff was coming from. He wasn't a physician. He wasn't a microbiologist in the Pasteur or Koch mold. He was a zoologist, a comparative embryologist, actually, someone who spent his days studying the development of simple organisms, invertebrates, creatures without blood or bones, or anything resembling a human immune system. And that's precisely why he could see what the medical researchers couldn't. Because he wasn't looking for germs. He wasn't thinking about disease at all. He was thinking about how organisms develop, how their cells differentiate and move and organize themselves. And when he saw those wandering cells in the starfish larva engulfing foreign matter, he didn't think interesting laboratory curiosity. He thought, this is how living things defend themselves. This is universal. He called these cells phagocytes, from the Greek words for eating and cell. And he spent the next several years building a case that phagocytosis, this process of cellular engulfment, was the primary mechanism of immunity, not just in starfish larvae, but in all animals, including humans. When you got an infection, Metchnikoff argued, your body's first and most important response was to send these roaming cells to the site of invasion, where they would surround and consume the invading bacteria. Inflammation, that redness, that swelling, that heat you feel around a wound, wasn't a sign that something was going wrong. It was a sign that something was going right. It was the battlefield. The phagocytes were rushing in, and the swelling was the result of that cellular army mobilizing. This was a genuinely revolutionary idea, and Metchnikoff pursued it with the kind of obsessive, almost manic energy that characterized his entire life. And his life, by the way, is one of the more dramatic stories in the history of science. He was born in 1845 in what's now Ukraine, showed extraordinary intellectual ability from childhood, published his first scientific paper at 16, and then spent much of his adult life battling severe depression. He attempted suicide at least twice, once by deliberately swallowing a large dose of opium, and another time by injecting himself with relapsing fever spirochetes, essentially trying to give himself a fatal infection. He survived both times, obviously, and the relapsing fever episode actually deepened his interest in how the body fights disease. There's something almost unbearably poignant about that. A man who tried to kill himself with a pathogen and then devoted the rest of his life to understanding how the body defends against pathogens. But anyway, the point is that Metchnikoff's phagocyte theory landed in the scientific world like a grenade, because by the 1880s and 1890s, there was already a competing explanation for how immunity worked, and it was coming out of the most powerful scientific institutions in Europe, the German laboratories, and the German school, led by researchers like Emil von Behring, Paul Ehrlich, and others working in or around Koch's orbit, had a very different answer to the question of how the body fights disease. Their answer wasn't cells, it was chemicals. Here's what happened. In 1890, Emil von Behring and a Japanese researcher named Kitasato Shibasaburo, working in Koch's laboratory in Berlin, made a discovery that would reshape medicine. They were studying diphtheria and tetanus, two horrific diseases, especially diphtheria, which was a leading killer of children in the 19th century. It attacked the throat, forming a thick membrane that could literally suffocate a child. Terrible disease. And what Behring and Kitasato found was this. If you took blood serum from an animal that had recovered from diphtheria and you injected that serum into another animal, the second animal became resistant to diphtheria too. Not permanently, but temporarily. Something in the blood, something dissolved in the liquid portion, the serum, with no cells involved, could transfer immunity from one animal to another. Think about what that means for a second. You didn't need to give the second animal a weakened version of the disease. You didn't need to train its body to fight. You just transferred some liquid, and suddenly it was protected. There was clearly something in that serum, some substance, some chemical factor, that was neutralizing the diphtheria toxin. Behring called it antitoxin. We would eventually call these substances antibodies, though that term came a bit later. This was not just a theoretical breakthrough. It was immediately, desperately practical. By 1891, Behring was using diphtheria antitoxin to treat sick children in Berlin. The results were dramatic, not perfect. The early preparations were crude and the dosing was inconsistent, but children who would have died were surviving. By the mid-1890s, diphtheria antitoxin was being produced on an industrial scale, often using horses as the source animals. You'd immunize a horse against diphtheria, wait for it to produce large quantities of antitoxin in its blood, then harvest the serum and inject it into sick patients. It was the first truly effective treatment for a major infectious disease, and Behring became a national hero for it. He won the very first Nobel Prize in Physiology or Medicine in 1901, largely for this work. And here's where the battle lines formed. Because if immunity was fundamentally about these soluble factors in the blood, these antitoxins, these antibodies, then what did you need Metchnikoff's phagocytes for? The German school, the humoralists as they came to be called, from the old medical term humors for bodily fluids, argued that the real action was chemical. Antibodies were what neutralized toxins. Antibodies were what marked bacteria for destruction. The cells were secondary, maybe even irrelevant. Metchnikoff naturally disagreed fiercely. He argued that the phagocytes were the primary defenders and that the soluble factors were at best helpers, substances that assisted the cells in doing their real work of engulfment and destruction. He had good evidence too. You could watch phagocytes eat bacteria under a microscope. You could see it happening. The chemical theory was more abstract, harder to visualize, and in some ways harder to prove mechanistically. But the German labs had institutional power, prestige, and, crucially, they had practical results. Antitoxin therapy was saving lives right now, today, in hospitals. Metchnikoff's cellular theory, however elegant, wasn't producing treatments. The rivalry became intensely personal. Metchnikoff felt marginalized, mocked by the German establishment. He eventually left Russia entirely and settled at the Pasteur Institute in Paris, where Pasteur, no friend of the Germans, as you might recall, gave him a laboratory and support. So now you had this fascinating geopolitical dimension layered onto the science. The French-Russian cellular camp versus the German humoral camp, with national pride and institutional funding, and career advancement all tangled up in what should have been a purely scientific question. And the thing is, both sides were right. That's what makes this story so instructive. Both sides were looking at real phenomena, real mechanisms that genuinely existed. The body does send phagocytic cells to engulf invaders. Mechnikov was absolutely correct about that. And the body does produce soluble antibodies that neutralize toxins and tag pathogens. Behring and Ehrlich were absolutely correct about that. The immune system uses both strategies simultaneously, and they're deeply intertwined. The antibodies that the humoralists discovered actually help the phagocytes that Mechnikov championed. Antibodies coat bacteria in a process called opsonization, which essentially makes the bacteria stickier, easier for phagocytes to grab and eat. It's like putting handles on a greased watermelon. The two systems aren't rivals. They're collaborators. But it took decades to work that out. Decades of arguments, of careers built on one camp and opposed to the other, of Nobel committees trying to split the difference. In 1908, the Nobel Prize in Physiology or Medicine was awarded jointly to Mechnikov and Ehrlich, one for the cellular theory, one for the humoral theory. A diplomatic solution, maybe, but also a tacit acknowledgement that the immune system was more complex than either camp had imagined. Now, let's talk about Paul Ehrlich for a moment, because he deserves more than a passing mention. Ehrlich is one of those figures in science who seems to have had about 4 separate careers, each of which would have been enough to make someone famous. Born in 1854 in what's now Poland, he was obsessed from an early age with chemical staining, the art of using dyes to color biological tissues so you could see them under a microscope. This might sound mundane, but it was transformative. Different dyes bind to different cellular structures, which means you can use them to reveal things that are otherwise invisible. Ehrlich's early work on staining led him to a crucial insight. If certain chemicals have an affinity for certain biological structures, then maybe you could find chemicals that bind specifically to disease-causing organisms and kill them without harming the patient. He called this idea a magic bullet, a term he actually coined, and it became the founding concept of chemotherapy. Not chemotherapy in the cancer sense, though that came later. Chemotherapy in the original sense, using chemicals to treat disease. Ehrlich's most famous magic bullet was salvarsan, developed around 1909, which was the first effective treatment for syphilis. It was an arsenic-based compound, yes, arsenic, that killed the syphilis spirochete while being tolerable, if not exactly pleasant, for the patient. It was the 606th compound Ehrlich tested, which is why it was sometimes called 606. 605 failures before one success. That's the reality of drug development, and it hasn't changed much. But for our purposes, Ehrlich's most important contribution was his side chain theory of antibody formation, proposed around 1897. Here's the idea, and it's beautifully intuitive. Ehrlich imagined that cells had chemical side chains on their surfaces, little molecular projections sticking out, each with a specific shape. When a toxin or pathogen came along with a matching shape, it would lock onto one of these side chains, like a key fitting into a lock. The cell, in response, would produce a flood of extra side chains, way more than it needed, and shed them into the bloodstream. Those shed side chains were the antibodies. They'd circulate freely, locking onto any matching toxin they encountered and neutralizing it. Now, Ehrlich got some of the details wrong. The mechanism of antibody production turned out to be more complicated than simple side chain shedding. But the core insight, that antibodies are specific, that each one matches a particular target, and that the body ramps up production of the right antibody when it encounters the right threat. That was essentially correct. And the lock and key metaphor he introduced became one of the most enduring images in all of biology. You've probably heard it used in contexts far beyond immunology. It shows up everywhere, from enzyme chemistry to neuroscience. That's Ehrlich's legacy, or at least one piece of it. So by the early 20th century, the picture was starting to come into focus, even if it was still blurry. The body had cellular defenders, phagocytes, that could engulf and destroy invaders. It also had chemical defenders, antibodies, that could neutralize toxins and mark pathogens for destruction. There was clearly some kind of specificity involved. The body could learn to recognize particular threats and respond more effectively the second time around. That was the whole basis of vaccination, going all the way back to Jenner and the milkmaids. But the mechanism of that learning, the actual biological process by which the body remembers a pathogen it encountered years ago, that remained deeply mysterious. And here's where the story takes a turn that nobody expected, because the next major breakthrough in understanding immunity didn't come from studying infections at all. It came from studying something that seemed completely unrelated: why some people's blood was compatible for transfusion and others wasn't. At the turn of the 20th century, blood transfusion was essentially a gamble. Sometimes it saved lives. Sometimes the recipient's blood would clump together, agglutinate in medical terminology, and they'd die horribly. Nobody knew why. The prevailing assumption was that blood was blood, that it was all basically the same stuff. But in 1901, a Viennese physician named Karl Landsteiner decided to systematically test what happened when you mixed blood samples from different people. And what he found was that blood was not all the same stuff, not even close. Landsteiner took blood samples from members of his laboratory, including himself, separated the red blood cells from the serum, and then methodically mixed each person's cells with each other person's serum. And he watched. Sometimes nothing happened. The cells floated around peacefully. Everything looked normal. But other times, the cells clumped together into visible clusters, sticking to each other in a way that was clearly abnormal. And the pattern wasn't random. It was consistent and repeatable. Certain combinations always clumped. Others never did. There was a logic to it. What Landsteiner realized was that people's blood fell into distinct groups. He initially identified 3. He called them A, B, and C, though C was later renamed O. His colleagues soon identified a 4th group, AB. And the rules were elegant. If you had type A blood, your red blood cells carried a particular molecule on their surface, an antigen, though that terminology came later. And your serum contained antibodies against the B molecule. If you had type B blood, it was the reverse. Type O had neither antigen on the cells, but antibodies against both A and B in the serum. And type AB had both antigens, but neither antibody. So when you mix type A cells with type B serum, the anti-A antibodies in that serum would latch onto the A antigens on those cells, cross-linking them, pulling them together into those deadly clumps. Think about what that means. The immune system wasn't just reacting to bacteria and viruses. It was reacting to molecules on the surface of human cells, cells from another person's body. The same basic mechanism Ehrlich had described, the lock and key specificity of antibodies recognizing antigens, was operating here too. But the target wasn't a pathogen. It was another person's tissue. This was the immune system drawing a line between self and not self, and it was drawing that line with extraordinary precision. Landsteiner published this work in 1901, the same year Behring was picking up his Nobel Prize for the diphtheria antitoxin. And honestly, at the time, Landsteiner's discovery didn't get the attention it deserved. It took years for the medical establishment to fully adopt blood typing before transfusions, even though the science was right there, clear as day. Landsteiner wouldn't receive his own Nobel Prize until 1930, nearly 3 decades later. But his work opened up a question that would dominate immunology for the next half-century. How does the body know what's self and what's foreign? How does it decide what to attack and what to leave alone? Because here's the thing. Your immune system is surrounded by your own cells every second of every day, trillions of them, and it doesn't attack them. It doesn't make antibodies against your own blood type antigens. It doesn't send phagocytes to devour your own liver cells. Somehow it has learned, or been taught, to recognize everything that belongs to you and to tolerate it, while remaining ready to pounce on anything that doesn't belong. And when that system breaks down, when the immune system does start attacking self, the results are devastating. We call those autoimmune diseases: rheumatoid arthritis, lupus, type 1 diabetes, multiple sclerosis. All of them are essentially the immune system making a terrible mistake, turning its weapons inward. But we're getting ahead of ourselves. In the early 1900s, autoimmunity was barely even a concept. Ehrlich himself had a term for it. He called it horror autotoxicus, which is a wonderfully dramatic name, the horror of self-toxicity. And he believed the body had built-in mechanisms to prevent it, that self-attack was essentially impossible under normal conditions. He was partly right and partly wrong, as it turned out, but the phrase stuck. So Landsteiner's blood groups established that immunological specificity extended far beyond infectious disease. The immune system could distinguish between incredibly similar molecules. The A antigen and the B antigen are chemically very close, just slightly different sugar chains on the surface of red blood cells. And yet the immune system treats them as completely different entities. That level of discrimination is remarkable. And Landsteiner spent much of his career pushing this further, testing whether antibodies could distinguish between synthetic chemicals that had never existed in nature. He would attach small chemical groups, called haptens, to proteins and inject them into animals, and sure enough, the animals would produce antibodies specific to those artificial molecules. Antibodies against chemicals that no living thing had ever encountered before in the history of life on Earth. This raised a staggering question. If the immune system could make specific antibodies against essentially any molecular shape, even ones that had never existed in nature, how was it doing that? How many different antibodies could the body potentially produce? The number seemed almost limitless. And yet antibodies are proteins, and proteins are encoded by genes, and genes are finite. There's only so much DNA in a human cell. This became known as the generation of diversity problem, and it haunted immunology for decades. How do you get an essentially infinite variety of antibodies from a finite genome? Now, there were 2 competing ideas about this. The first was called the instructive theory, and it seemed like the most intuitive explanation. The idea was that antibodies were basically flexible templates. When a foreign antigen entered the body, it would physically mold the antibody around itself, like pressing a key into soft wax to make an impression. The antigen instructed the antibody's shape. This was proposed in various forms by several scientists, including Linus Pauling. Yes, that Linus Pauling, the double Nobel laureate, one of the most brilliant chemists of the 20th century. In 1940, Pauling suggested that antibodies were all made from the same basic protein, and they acquired their specificity by folding around the antigen they encountered. It was an elegant idea. It made intuitive sense, and it was completely wrong. The alternative was called the selective theory, and its modern form was proposed in 1957 by an Australian virologist named Frank McFarlane Burnett. Burnett's idea, called the clonal selection theory, was radical, and it changed everything. Here's what he proposed. Instead of antibodies being molded by antigens, Burnett said that the body pre-makes an enormous variety of immune cells, each one carrying a unique, randomly generated receptor on its surface. Each cell can only recognize one specific molecular shape. Before you ever encounter a pathogen, before you're ever exposed to any foreign substance, your body has already produced millions of different immune cells, each one specific to a different antigen. Most of them will never encounter their matching antigen. They'll live and die without ever being activated. But when a pathogen does enter your body, and it happens to bump into the one cell, out of millions, whose receptor matches one of the pathogen's surface molecules, that cell gets activated. It starts dividing rapidly, making clones of itself. A whole army of identical cells, all specific to that one antigen. That's the clonal part. The selection happens because the antigen selects which clone gets expanded. The antigen doesn't shape the antibody. The antibody was already there, waiting. The antigen just finds it. Think about what a different picture that paints. Your immune system isn't a blank slate that learns on the job. It's more like a library that already contains a book about every possible topic, most of which nobody will ever read. But when you need one specific book, you can find it, pull it off the shelf, and print 1 million copies. The information was always there. It just needed to be selected. And here's where it connects back to immunological memory, the thing Thucydides noticed during the plague of Athens 2,500 years ago. When that activated cell divides and makes its army of clones, most of those clones become effector cells. They go to war. They fight the infection, and when the battle's over, they die. But some of them become memory cells. They persist in your body for years, sometimes decades, sometimes for the rest of your life, and they remain specific to that same antigen. So if the same pathogen shows up again, 5 years later, 10 years later, those memory cells are already there. They don't need to be selected from scratch. They activate faster, they multiply faster, they produce antibodies faster and in greater quantities. That's why the second infection is milder or doesn't happen at all. That's why vaccines work. You're essentially pre-selecting the right clone and building a stockpile of memory cells, all without having to suffer through the actual disease. Burnett published his clonal selection theory in 1957, and it was beautiful in its logic, but it was still a theory. It needed evidence, and the evidence came from multiple directions over the following years. One crucial piece came from Gustav Nossal and Joshua Lederberg, who demonstrated in 1958 that individual antibody-producing cells really do make only one type of antibody. One cell, one antibody, exactly as Burnett's theory predicted. Another piece came from James Gowans in Oxford, who showed in the early 1960s that lymphocytes, a type of white blood cell that had been somewhat neglected, were actually the key players in adaptive immunity. Gowans demonstrated that if you drained the lymphocytes from a rat, its immune system essentially collapsed. Put them back, and immunity returned. Lymphocytes were the cells carrying those pre-made receptors, the ones being selected and cloned. And this is where the picture starts to get really intricate, because it turns out there isn't just one kind of lymphocyte. There are 2 major types, and they do very different things. And the story of how we figured that out involves some of the most clever experiments in the history of biology. The 2 types are B cells and T cells. You've almost certainly heard those terms, especially if you lived through the COVID pandemic. But the distinction between them and the discovery of their separate roles took decades of painstaking work. The B cell story starts, oddly enough, with chickens. In the 1950s and early 1960s, Researchers studying chickens noticed that these birds had a peculiar organ called the bursa of Fabricius, a small pouch near the tail end of the gut. If you removed this organ from a young chick, something strange happened. The chicken could still reject skin grafts, a type of immune response, but it couldn't produce antibodies. It lost its humoral immunity while retaining its cellular immunity. That was a stunning finding because it suggested that the two arms of the immune response, the ones that Metchnikoff and Ehrlich had argued about so bitterly, were actually controlled by different populations of cells. The antibody-producing cells depended on the bursa. They became known as B cells, B for bursa. But mammals don't have a bursa, so where do our B cells come from? It turned out they mature in the bone marrow, which conveniently also starts with B, so the name worked out. But the T cells, that's a different story entirely, and it leads us to one of the strangest and most underappreciated organs in your body. An organ that most people have never thought about, that sits right behind your breastbone, that's largest when you're a child and slowly shrinks as you age, and that turned out to be absolutely essential for the most sophisticated branch of your immune system. It's called the thymus, and for a long time, nobody had any idea what it did. Anatomists had known about the thymus for centuries. Galen described it way back in the 2nd century, but it was considered a vestigial organ, a leftover, something the body didn't really need. And you can understand why people thought that. If you look at the thymus in an adult, it's small, fatty, kind of unremarkable. In older adults, it's barely distinguishable from the surrounding tissue. It doesn't look like it's doing much of anything, and if you removed it from an adult animal, nothing obviously terrible happened. The animal seemed fine. So for a long time, the thymus was one of those anatomical curiosities that nobody paid much attention to. But then Jacques Miller changed everything. Miller was a young Australian researcher, born in France actually, who in the early 1960s was working at the Chester Beatty Research Institute in London. He was studying mouse leukemia, which was known to involve a virus that affected the thymus. And he decided to try something that, in retrospect, seems almost too simple. He removed the thymus from newborn mice. Not adult mice, where the operation didn't seem to matter much, but mice that were just hours old. And what happened was dramatic. These neonatally thymectomized mice, that's the technical term, but really, it just means baby mice with their thymus taken out. They were devastated immunologically. They couldn't reject skin grafts. You could take skin from a completely unrelated mouse, even from a rat, graft it onto one of these thymectomized mice, and the graft would just sit there, accepted, as if the mouse's immune system couldn't recognize it as foreign. They were also profoundly susceptible to infections. They wasted away. Their lymph nodes were depleted of a particular population of small lymphocytes. Miller published his findings in 1961 and 1962. And here's the thing. The scientific community was skeptical. Some people just didn't believe it. The thymus, that useless little lump, essential for immunity, it seemed too strange. Sir Peter Medawar, who had won the Nobel Prize in 1960 for his work on immunological tolerance, we'll come back to him, reportedly said that the thymus was about as useful as the appendix, but Miller's results were solid, and they were replicated, and gradually the picture came into focus. The thymus was the organ where T cells matured. That's where the T comes from, T for thymus, just as B is for bursa or bone marrow. The thymus took immature precursor cells that originated in the bone marrow and turned them into functional T cells. And without that process, without that education in the thymus, the entire cellular arm of adaptive immunity collapsed. Now, think about what that means in terms of the old debate between Metchnikoff and Ehrlich. Remember how they argued about whether immunity was primarily cellular, about the eating cells, the phagocytes, or humoral, about the soluble factors in the blood, the antibodies? And remember how the resolution was that both were important, that they worked together? Well, now the picture was getting even more refined. It wasn't just that cellular and humoral immunity coexisted. They were actually run by two distinct lineages of lymphocytes, B cells and T cells, that matured in different organs and performed fundamentally different jobs. B cells made antibodies. T cells did something else, something that turned out to be far more complex and varied than anyone initially imagined. So what do T cells actually do? Let's talk about that, because this is where immunology gets genuinely intricate and also genuinely beautiful. T cells come in several major flavors, but the two you hear about most are helper T cells and killer T cells. Their formal names are CD4 positive and CD8 positive T cells, named for surface molecules they carry, but helper and killer capture their roles pretty well. Killer T cells, also called cytotoxic T lymphocytes, do exactly what the name suggests. They kill other cells, but not just any cells. They kill your own cells that have become dangerous. Cells that are infected by viruses, for instance, or cells that have turned cancerous. And here's what's remarkable about how they do it. A killer T cell doesn't just wander around looking for trouble. It's looking for something very specific. It's scanning the surfaces of your cells, checking the molecular display that every nucleated cell in your body presents on its surface. This display system is called the Major Histocompatibility Complex, or MHC. And understanding MHC is one of those things that, once you grasp it, makes the whole immune system click into place in a new way. Think of it like this. Every cell in your body is constantly doing stuff, making proteins, breaking them down, carrying out its normal functions. As part of that process, cells take little fragments of the proteins they're producing, small peptide snippets, and they display them on their surface, held in the groove of an MHC molecule, almost like a hot dog sitting in a bun. These fragments are presented outward, facing the extracellular world, as if the cell is holding up a little sign that says, "Here's what I'm making right now." Under normal circumstances, all those fragments are pieces of your own normal proteins, and T cells recognize them as self and move on. Nothing to see here. But if a virus has infected that cell, something changes. Now the cell is producing viral proteins because the virus has hijacked the cell's machinery to replicate itself. And fragments of those viral proteins get loaded onto MHC molecules and displayed on the surface too. So now the cell's little sign reads differently. It's showing foreign peptides, and a killer T cell whose receptor happens to match that particular foreign fragment. Remember Burnett's clonal selection. One cell, one specificity. That T cell recognizes the infected cell and destroys it. It releases molecules called perforin and granzymes that punch holes in the target cell's membrane and trigger it to self-destruct through apoptosis, which is programmed cell death. The infected cell dies, taking the virus inside it down too. This is fundamentally different from what antibodies do. Antibodies work in the fluid spaces, the blood, the lymph, the mucous membranes. They're great at neutralizing free-floating pathogens, things that are outside of cells. But viruses, once they get inside a cell, are invisible to antibodies. They're hiding behind the cell membrane. Killer T cells solve that problem by essentially performing quality control on every cell in the body, checking what each cell is making and eliminating any cell that's been compromised. It's an internal surveillance system, and it's extraordinarily precise. Now, helper T cells do something different and arguably even more important. They're the coordinators, the conductors of the immune orchestra, if you want a metaphor. When a helper T cell encounters its matching antigen, presented to it by special cells called antigen-presenting cells, which include those macrophages that Mechnikov loved so much, as well as dendritic cells and B cells. When a helper T cell gets activated, it doesn't go out and kill anything directly. Instead, it releases signaling molecules called cytokines that amplify and direct the immune response. It helps B cells produce better antibodies. It helps killer T cells become more lethal. It activates macrophages to become more aggressive phagocytes. It shapes the entire character of the immune response, tuning it to match the type of threat. And this is why the loss of helper T cells is so catastrophic. Which brings us to one of the darkest chapters in immunological history, and one that unfolded in real time in front of the world, starting in the early 1980s. In June of 1981, the CDC published a short report about 5 young men in Los Angeles who had developed pneumocystis carinii pneumonia, a rare lung infection caused by a fungus that almost never affected people with healthy immune systems. All 5 men were previously healthy. All 5 were gay. Within weeks, more cases appeared, not just pneumocystis, but Kaposi's sarcoma, a rare cancer, and other opportunistic infections that doctors typically only saw in patients whose immune systems had been deliberately suppressed, like organ transplant recipients on immunosuppressive drugs. Something was destroying these patients' immune systems. And when researchers looked at their blood, they found the answer, or at least the proximate answer. These patients had dramatically reduced numbers of helper T cells. Their CD4 counts, which in a healthy person run between roughly 500 and 1,500 cells per microliter of blood, were plummeting towards 0. Without helper T cells to coordinate the immune response, everything fell apart. The killer T cells didn't get properly activated. The B cells couldn't produce effective antibodies. The macrophages weren't ramped up. The entire adaptive immune system was essentially decapitated. The generals had been killed, and the army was in disarray. The disease became known as AIDS, acquired immunodeficiency syndrome, and the virus that caused it, identified in 1983 by Luc Montagnier's group at the Pasteur Institute in Paris, and independently by Robert Gallo's lab at the NIAID in the United States, was eventually named HIV, the human immunodeficiency virus. And the bitter priority dispute between Montagnier and Gallo over who really discovered the virus first became one of the most acrimonious scientific controversies of the 20th century, echoing, in a way, the old Pasteur-Koch rivalry, complete with national pride, accusations of misconduct, and eventual diplomatic resolution. Both Montagnier and his colleague, Françoise Barré-Sinoussi, received the Nobel Prize in 2008. Gallo did not, which tells you something about how the scientific community ultimately judged the matter, though the full story is more nuanced than any prize can capture. But the immunological lesson of HIV is what matters for us tonight. HIV specifically targets helper T cells. It binds to the CD4 molecule on their surface. That's its entry point, its doorway into the cell. Once inside, it integrates its genetic material into the T cell's DNA and turns the cell into a virus factory, eventually killing it. And because the virus specifically destroys the cells that coordinate the entire adaptive immune response, the result is a progressive, systematic dismantling of immunity. It's as if someone figured out exactly which wire to cut to bring down the whole electrical grid. The elegance of it, from the virus's perspective, is terrifying. And the suffering it caused, has caused, continues to cause, is immense. More than 40 million people have died of AIDS-related illnesses since the epidemic began. HIV taught the world, in the most painful way possible, just how central helper T cells are to everything the immune system does. And it also drove an enormous wave of immunological research. Funding poured in. Thousands of scientists redirected their careers. The detailed molecular understanding of how T cells work, how they recognize antigens, how they signal to each other, how they develop in the thymus. Much of that knowledge was accelerated by the urgent need to understand what HIV was doing and how to stop it. And that urgency led researchers deeper into the thymus, into the remarkable process by which T cells are educated. Because here's the thing about T cells that makes them both incredibly powerful and incredibly dangerous. They have the potential to attack your own tissues. Remember Ehrlich's horror autotoxicus, the body's supposed horror of self-destruction? The immune system needs T cells that can recognize virtually any foreign protein, any pathogen that might come along. But it also needs to make absolutely sure those T cells don't attack the body's own proteins. And that's a razor-thin line to walk, because the receptors on T cells are generated randomly through a process of gene rearrangement that we haven't fully gotten into yet, but that produces an astonishing diversity of specificities. Some estimates suggest your body can generate T cell receptors capable of recognizing something on the order of 10 to the 15 or even 10 to the 18 different molecular shapes. That's a number so large it's essentially meaningless to try to visualize. But among all those randomly generated receptors, some of them, purely by chance, will be perfectly shaped to recognize your own normal proteins. And if those self-reactive T cells escape into the body, they'll attack your own tissues. That's autoimmunity. That's what Ehrlich feared. So the thymus runs a brutal screening process. Immature T cells enter the thymus from the bone marrow, and they go through what immunologists sometimes call thymic education, though education makes it sound gentler than it is. It's more like an audition where the penalty for failure is death. The process has two main stages. In positive selection, T cells are tested to make sure their receptors can actually interact with MHC molecules, because if a T cell can't read the MHC display system, it's useless. T cells that fail this test die by neglect. They simply don't receive the survival signals they need. In negative selection, the surviving T cells are then tested against self-antigens, proteins from your own body that are presented in the thymus. If a T cell's receptor binds too strongly to a self-antigen, meaning it would likely attack your own tissues if released, that T cell is eliminated. It's induced to undergo apoptosis right there in the thymus, killed before it can do any harm. The numbers here are staggering. Something like 95 to 98% of all T cells that enter the thymus die there. They fail one test or the other. Only that tiny surviving fraction, the cells that can interact with MHC well enough to be functional, but that don't react too strongly against self, only those cells graduate and enter the bloodstream as mature, naive T cells, ready to encounter their matching antigen for the first time out in the real world. And there's a beautiful detail about how the thymus manages to test T cells against self-antigens from all over the body. You might wonder, the thymus is just one small organ sitting behind the breastbone. How can it possibly present proteins from your liver, your brain, your pancreas, your skin? It's not connected to all those tissues. The answer involves a remarkable gene called AIRE, autoimmune regulator. Cells in the thymus, called medullary thymic epithelial cells, turn on this gene, and what AIRE does is almost magical. It allows these thymic cells to express proteins that would normally only be made in specific distant tissues. Insulin, for example, which is normally only made by beta cells in the pancreas, or proteins specific to the lens of the eye, or thyroid-specific proteins. AIRE forces the thymic cells to produce a sampling of these tissue-restricted proteins so that developing T cells can be tested against them. If a T cell reacts to insulin presented in the thymus, it gets deleted, and that T cell never reaches the pancreas to cause trouble. When AIRE doesn't work, when the gene is mutated, the consequences are devastating, and they tell you exactly how important this process is. There's a rare human disease called autoimmune polyendocrinopathy syndrome, or APS1, caused by mutations in AIRE. People with this condition develop autoimmune attacks against multiple organs: their adrenal glands, their parathyroid glands, their skin, their liver. Their immune system turns on their own body because the thymic screening process was incomplete, because the T cells that should have been eliminated were never tested against those tissue-specific proteins and slipped through into circulation. This whole system, the generation of diversity, the thymic education, the clonal selection of the survivors. It's one of the most elegant solutions to an engineering problem you'll ever encounter. The problem is, how do you build a defense system that can recognize threats you've never seen before, that might not even exist yet, while simultaneously making sure it doesn't destroy the very thing it's supposed to protect? And the solution is to generate diversity randomly, screen ruthlessly, and then let the environment select what's needed. It's almost Darwinian in its logic: variation, selection, amplification. And in fact, Burnett explicitly drew that parallel when he proposed clonal selection theory. He saw the immune system as evolution in miniature, playing out inside your body over days rather than millennia. But the screening isn't perfect. Some self-reactive T cells do escape the thymus, and that's where things get really interesting, because the immune system has backup mechanisms, layers of tolerance beyond thymic deletion. And when those backups fail, you get autoimmune diseases that affect millions of people. Diseases like type 1 diabetes, where T cells attack the insulin-producing beta cells of the pancreas, or multiple sclerosis, where they attack the myelin sheath around nerve fibers, or rheumatoid arthritis, where they attack the lining of the joints. These are not rare curiosities. They are among the most common chronic diseases in the developed world, and understanding them requires understanding those backup mechanisms, the ones that operate outside the thymus, in the periphery of the body, where mature T cells are patrolling and working, and occasionally, dangerously, making mistakes. The most fascinating of these peripheral tolerance mechanisms involves a special subset of T cells that doesn't attack anything at all. Instead, its entire job is to suppress other T cells, to keep the immune system in check, to prevent exactly the kind of self-destructive overreaction that leads to autoimmune disease. These cells are called regulatory T cells, and for a long time, almost nobody believed they existed. That's not an exaggeration. The idea that there might be a class of T cells whose job was to actively suppress immune responses was floated back in the early 1970s by a researcher named Richard Gershon at Yale. Gershon called them suppressor T cells, and he published a series of experiments suggesting that certain T cells could dampen the activity of other immune cells. It was a genuinely interesting idea, and for a while, it gained traction. But here's what went wrong. The field couldn't find a reliable molecular marker for these cells. Nobody could pin down exactly which T cells were doing the suppressing, or how. The experiments were hard to reproduce, and as immunology matured through the late 70s and into the 80s, the whole concept of suppressor T cells fell into disrepute. It became almost a dirty word in immunology. If you brought up suppressor T cells at a conference in the late 1980s, people would look at you the way engineers might look at someone proposing perpetual motion. It was considered a dead end, an artifact of sloppy experiments. Gershon himself died in 1983 before any vindication came, and the field moved on. Except the problem didn't go away. Because the thymus, as thorough as it is, can't catch everything. Remember, roughly 2 to 5% of T cells survive thymic selection, and among those survivors, some inevitably carry receptors that can recognize self-antigens, just not strongly enough to trigger deletion during negative selection. They slip through. They're out there in your blood, in your lymph nodes, in your tissues. And yet most people don't develop autoimmune disease. Something is keeping those potentially dangerous T cells in check, something beyond the thymus. The breakthrough came in the mid-1990s from a Japanese immunologist named Shimon Sakaguchi. Working at Kyoto University, Sakaguchi identified a population of CD4 positive T cells that constitutively expressed a surface molecule called CD25, which is part of the receptor for a signaling molecule called interleukin-2. Now, CD25 by itself wasn't unique. Activated T cells express it too. But Sakaguchi showed that if you took these CD4 positive, CD25 positive cells out of a mouse, the mouse developed devastating autoimmune disease, inflammation of the thyroid, the stomach, multiple organs under attack. And if you put those cells back in, the autoimmune disease was prevented. It was a clean, elegant experiment, and it reopened the entire question that Gershon had raised 2 decades earlier. These were real. Suppressor cells existed. The field just hadn't had the tools to find them properly. Sakaguchi's work was published in 1995, and it changed immunology. The cells were eventually renamed regulatory T cells, TREGs for short, partly to distance them from the baggage of the old suppressor T cell concept, and partly because the new name better captured what they actually do. They regulate, they modulate. They're not just an off switch, they're more like a thermostat, constantly adjusting the intensity of immune responses to keep everything in a safe range. And here's what's really clever about how they work. TREGs use multiple mechanisms to suppress other immune cells. Some of these are direct. A TREG can physically interact with a T cell that's becoming activated and essentially tell it to stand down through surface molecules like CTLA4 that compete for the same signals that T cells need to get fully activated. Some mechanisms are indirect. TREGs secrete anti-inflammatory cytokines, signaling molecules like interleukin-10 and TGF-beta that create a local environment of calm, dampening the activity of nearby immune cells. And some mechanisms are almost sneaky. TREGs express such high levels of the interleukin-2 receptor that they essentially soak up IL-2 from the local environment, starving other T cells of a growth signal they need to proliferate. It's like someone at a buffet who takes all the food before anyone else can get to the table. The other T cells don't get the signal to multiply, so the response fizzles out. Think about what that means for your body right now. At this very moment, you have regulatory T cells patrolling your tissues, keeping watch not over invaders, but over your own immune system. They're the peacekeepers, and when they fail, when their numbers drop or their function is impaired, the consequences can be severe. There's a rare genetic condition called IPEX syndrome, which stands for immune dysregulation, polyendocrinopathy, enteropathy, X-linked. It's caused by mutations in a gene called FOXP3, which turns out to be the master transcription factor that defines TREG identity. Without functional FOXP3, you essentially don't make proper regulatory T cells, and children born with IPEX develop overwhelming autoimmune disease affecting multiple organs, the gut, the skin, the endocrine glands. Without treatment, it's often fatal in early childhood. It's a heartbreaking condition, but it taught immunologists something profound. FOXP3 is not just a marker for Tregs, it's the gene that makes a Treg a Treg. Without it, the whole regulatory arm of adaptive immunity collapses. So now you can see the full architecture of self-tolerance, and it really is layered like a well-designed safety system. Central tolerance in the thymus handles the most obviously dangerous T cells. The ones with strong self-reactivity get deleted. The AIRE gene makes sure that even tissue-specific proteins are represented during that screening process. But the cells that escape with low-level self-reactivity are then managed in the periphery by Tregs, by anergy, a state where a T cell encounters its antigen but doesn't receive the right co-stimulatory signals and becomes functionally unresponsive. And by other mechanisms we're still mapping out. It's defense in depth. Multiple independent systems, each catching what the others miss. And this brings us to something that connects all of this to one of the most important medical frontiers of our time: cancer immunology. Because here's the uncomfortable truth about Tregs and peripheral tolerance. The same mechanisms that prevent autoimmune disease can also prevent your immune system from attacking tumors. Think about that for a second. A cancer cell is, fundamentally, one of your own cells that has gone wrong. Its DNA has accumulated mutations, and it's growing without the normal controls. But it's still expressing many of the same surface proteins as your healthy cells. From the immune system's perspective, a tumor can look an awful lot like self. And all those tolerance mechanisms we just talked about, the ones designed to prevent attacks on self, can actually protect the tumor. Tregs accumulate in the tumor microenvironment, secreting their calming cytokines, soaking up IL-2, telling killer T cells to stand down. The tumor itself often exploits these pathways, actively recruiting Tregs and expressing molecules that engage the immune system's brakes. This is where a concept called immune checkpoints comes in, and it's a story that earned Jim Allison and Tasuku Honjo the Nobel Prize in Physiology or Medicine in 2018. Allison, working at the University of California, Berkeley, and later MD Anderson Cancer Center, had been studying a molecule called CTLA-4 since the early 1990s. CTLA-4 is one of those brake molecules. It's expressed on T cells, and when it engages its ligand, it sends a signal that dampens T cell activation. It's part of the normal regulatory machinery, but Allison had a radical idea. What if you blocked CTLA-4? What if you released the brake and let T cells attack the tumor? He tested this in mice in the mid-1990s, using antibodies that blocked CTLA-4, and the results were striking. Tumors that would normally grow unchecked were rejected by the immune system once the checkpoint was blocked. The T cells were there all along. They could see the tumor, but they were being held back by the very tolerance mechanisms that normally prevent autoimmunity. Remove the brake, and suddenly the immune system does what it's supposed to do. Meanwhile, in Kyoto, Tasuku Honjo had discovered another checkpoint molecule called PD-1, programmed Death 1, back in 1992. PD-1 operates through a slightly different mechanism than CTLA-4, but achieves a similar result. It tells T cells to stop attacking. Tumors, it turned out, were particularly good at exploiting PD-1. Many cancer cells express a ligand called PDL1 on their surface, which binds to PD-1 on incoming T cells and essentially says, "Nothing to see here, move along." It's molecular camouflage, using the immune system's own tolerance machinery as a shield. The clinical development of checkpoint inhibitors, antibodies that block CTLA-4 or PD-1 or PDL1, has been one of the most dramatic stories in modern medicine. Ipilimumab, the anti-CTLA-4 antibody, was approved for metastatic melanoma in 2011, for a disease that had been essentially untreatable in its advanced stages. Some patients were now experiencing durable remissions lasting years. Not everyone responded, that's important to say, but for those who did, it was transformative. Anti-PD-1 drugs like nivolumab and pembrolizumab followed, and they've been effective across a wider range of cancers: lung cancer, kidney cancer, bladder cancer, certain types of lymphoma. But here's the thing that ties this back to everything we've been talking about. The side effects of checkpoint inhibitors are, predictably, autoimmune in nature. You're releasing the brakes on the immune system. You're disabling the very mechanisms that prevent self-attack. So patients on these drugs can develop inflammation of the colon, the liver, the skin, the thyroid, the pituitary gland. It's the price of unleashing the immune system against cancer. You also risk unleashing it against yourself. It's the fundamental tension at the heart of immunology, the same tension that Ehrlich intuited when he coined horror autotoxicus over a century ago. The immune system's power and its danger are two sides of the same coin. And this tension doesn't just play out in cancer therapy. It shows up everywhere in modern medicine, in organ transplantation, where you have to suppress the immune system to prevent rejection of a donated organ, which then leaves the patient vulnerable to infections and cancers. In allergies, where the immune system mounts an aggressive response against something completely harmless, like pollen or peanut protein. In chronic inflammatory diseases, where the immune system's response to a perceived threat causes more damage than the threat itself. Each of these is a story about calibration, about a system that has to be aggressive enough to protect you, but restrained enough not to destroy you. And the engineering of that balance is breathtakingly complex. Which brings us to something I've been wanting to talk about. The part of the immune system that doesn't get nearly enough credit. The part that was working long before your adaptive immune system ever got involved. Long before T cells and B cells and antibodies. The innate immune system. We touched on Mechnikov and his phagocytes earlier, but there's so much more to this story, and the discoveries of the late 20th century revealed that innate immunity isn't just a blunt, primitive first line of defense. It's actually the system that decides whether the adaptive immune system gets activated at all, and how it gets activated, and what kind of response it mounts. The innate immune system is the gatekeeper, the intelligence service, the one reading the battlefield and making the strategic calls. And the person who cracked open that understanding was a researcher named Charles Janeway, working at Yale in the late 1980s, who posed a question so simple it was almost embarrassing that nobody had asked it before. The question was this: how does the immune system know the difference between something foreign that's dangerous and something foreign that's not dangerous? Because think about it. Your body encounters foreign material all the time. Every time you eat a meal, your gut is flooded with foreign proteins. Every breath you take pulls in dust particles, fungal spores, bits of dead skin from other people. Most of it is completely harmless, but some of it, a pathogenic bacterium, a virus, a parasitic organism, is genuinely threatening. And somehow, the immune system has to tell the difference. Not eventually, not after days of deliberation, but fast, within minutes. Because if a dangerous pathogen gets a foothold and you wait around for the adaptive immune system to spin up its slow, precise, antibody-generating machinery, you could be dead before the cavalry arrives. Janeway called this the immunologist's dirty little secret. And the secret was this: for decades, immunologists had been so focused on the adaptive immune system, on the exquisite specificity of antibodies and T-cell receptors, on clonal selection and immunological memory, that they'd mostly ignored a fundamental question about how the whole thing gets started. See, your adaptive immune system doesn't just spontaneously activate. It needs a signal. It needs something to tell it, hey, wake up, there's a real threat here. Mount a response. And that signal, Janeway argued, had to come from the innate immune system. The older system. The ancient one. The one that evolved hundreds of millions of years before adaptive immunity ever appeared. Here's what Janeway proposed, and it was elegant. He said the innate immune system must have receptors. He called them pattern recognition receptors that don't recognize specific antigens the way antibodies do. They don't care about the particular shape of one protein on one strain of one bacterium. Instead, they recognize broad molecular patterns that are shared by whole classes of pathogens. Patterns that are essential to the pathogen's survival, so they can't easily mutate away from them. Patterns that are fundamentally different from anything found on human cells. He called these pathogen-associated molecular patterns, or PAMPs. And the beauty of this idea is that it solves the problem of speed and breadth simultaneously. You don't need millions of different receptors to cover every possible pathogen. You need a relatively small set of receptors that recognize the molecular signatures common to bacteria as a group, or viruses as a group, or fungi as a group. Things like lipopolysaccharide, which is a component of the outer membrane of gram-negative bacteria, or double-stranded RNA, which is a hallmark of viral replication. Human cells don't normally have double-stranded RNA floating around, but many viruses produce it when they're copying themselves. Or flagellin, the protein that makes up bacterial flagella. These are molecular calling cards that scream pathogen to the innate immune system. Janeway presented this idea at a Cold Spring Harbor symposium in 1989, and honestly, the reception was lukewarm. A lot of immunologists were so deep in the adaptive immunity paradigm that this seemed almost too simple, too old-fashioned, like going back to talk about phagocytes when everyone wanted to talk about T-cell receptor gene rearrangement. But Janeway was right, and the proof came about a decade later through a series of discoveries that connected his theoretical framework to actual physical molecules. The breakthrough came from an unexpected direction: fruit flies. In 1996, a researcher named Jules Hoffmann, working in Strasbourg, France, was studying the immune system of Drosophila. Now, fruit flies don't have an adaptive immune system. No antibodies, no T cells, no B cells. They rely entirely on innate immunity. And Hoffman's group discovered that a gene called TOL, which had originally been identified for its role in embryonic development, was also critical for the fly's ability to fight off fungal infections. Flies with mutations in the TOL gene couldn't mount an effective antifungal response, and they died from infections that normal flies shrugged off easily. The TOL receptor, it turned out, was recognizing something about the fungal pathogen and triggering a defensive response. This was a huge clue, and it sent researchers scrambling to find the mammalian equivalent. The person who found it was Bruce Beutler, working at the Scripps Research Institute in La Jolla, California. In 1998, Beutler identified a mammalian version of the TOL receptor. He called it Toll-like receptor 4, or TLR4, and showed that it was the receptor responsible for recognizing lipopolysaccharide, that bacterial membrane component I mentioned. This was the receptor that, when it detected LPS, triggered the inflammatory cascade that you experience as the redness, swelling, heat, and pain of an infection. Mice with a mutation in TLR4 couldn't respond properly to gram-negative bacteria. They were sitting ducks. And here's what's really satisfying about this. It was exactly what Janeway had predicted. A pattern recognition receptor on innate immune cells, recognizing a conserved molecular pattern shared by a whole class of pathogens, not one specific bacterium. All gram-negative bacteria, one receptor, millions of potential threats covered. Hoffman and Beutler shared the Nobel Prize in 2011 for these discoveries, and Janeway almost certainly would have shared it too, but he had passed away from cancer in 2003. One of those bittersweet stories in science where the person who saw the answer most clearly didn't live to see the full recognition. After TLR4, researchers found a whole family of toll-like receptors in humans, about 10 of them, each recognizing different pathogen-associated patterns. TLR3 recognizes double-stranded RNA from viruses. TLR5 recognizes bacterial flagellin. TLR9 recognizes unmethylated CpG DNA, which is common in bacterial genomes but rare in vertebrate genomes, and they're positioned strategically throughout the body. Some sit on the outer surface of cells that patrol the tissues, macrophages, dendritic cells, the sentinels of the innate immune system. Others are tucked inside the cell, in compartments called endosomes, where they can detect viral genetic material that's been swallowed up during phagocytosis. It's a surveillance network, distributed and layered, each receptor tuned to a different frequency of danger. But the toll-like receptors turned out to be just one family among several. Researchers discovered other classes of pattern recognition receptors, NOD-like receptors that patrol the interior of the cell, RIG-I-like receptors that detect viral RNA in the cytoplasm, C-type lectin receptors that recognize sugar structures on the surfaces of fungi. The innate immune system, far from being the blunt, unsophisticated system it had been dismissed as, turned out to have a remarkably nuanced sensory apparatus. It can distinguish between bacteria and viruses, between fungi and parasites, and it tailors the initial response accordingly. And here's the crucial part. It tells the adaptive immune system what kind of threat it's dealing with. This is the gatekeeper function that Janeway envisioned. When a dendritic cell in your skin encounters a bacterium, its toll-like receptors recognize the bacterial patterns, and the cell becomes activated. It swallows the bacterium, chops its proteins into peptides, and loads those peptides onto MHC molecules, the same molecular display cases we talked about earlier. But here's what makes the innate recognition essential. The dendritic cell also upregulates co-stimulatory molecules on its surface. These are the second signal. Remember how we said T cells need two signals to become activated: the antigen presented on MHC plus a co-stimulatory signal. Without that second signal, the T cell doesn't activate. It becomes allergic, tolerant, unresponsive, and it's the innate immune system's pattern recognition receptors that control whether that second signal gets delivered. Think about what that means. If a dendritic cell encounters a foreign protein, but its pattern recognition receptors don't detect any danger signals, no bacterial components, no viral RNA, no signs of tissue damage, then it presents the antigen without co-stimulation, and the T cell that recognizes it is shut down rather than activated. This is one of the reasons your immune system doesn't attack the food proteins in your gut or the harmless bacteria that live on your skin. Foreign, yes. Dangerous, no. No danger signals, no co-stimulation, no adaptive immune response. It's an incredibly elegant solution to the problem Janeway identified. And it gets even more interesting when you consider what happens during tissue damage, even in the absence of infection. In the early 2000s, a researcher named Polly Matzinger at the National Institutes of Health had been pushing a complementary idea she called the danger model. Matzinger argued that the immune system doesn't really care about self versus non-self per se. What it really cares about is danger. And she pointed out that when your own cells are damaged or dying in a traumatic, uncontrolled way, crushed in an injury, killed by a toxin, destroyed by a burn, they release their internal contents into the surrounding tissue. Molecules that are normally safely tucked inside cells suddenly flood the extracellular space. Things like uric acid, ATP, certain heat shock proteins, fragments of mitochondrial DNA. These molecules, which came to be called damage-associated molecular patterns, or DAMPs, can also trigger pattern recognition receptors on innate immune cells, your own molecules, acting as alarm signals. This explains something that had puzzled immunologists for a long time: why the immune system responds to sterile injuries, situations where there's no pathogen at all. You slam your finger in a door, and it swells up, turns red, gets hot and painful. That's inflammation. That's your innate immune system responding. But responding to what? There's no bacterium. There's no virus. The answer is that the crushed cells released their DAMPs, and the innate immune system interpreted that as danger, which makes sense from an engineering perspective. If tissue is damaged, there's a good chance a pathogen could exploit that breach. So mounting a preemptive inflammatory response is a reasonable defensive strategy. So now you have this picture of the innate immune system as something far more sophisticated than anyone appreciated for most of the 20th century. It's reading the environment constantly, using dozens of different pattern recognition receptors to detect both pathogen-associated and damage-associated molecular patterns. It's making judgment calls in real time. Is this dangerous? What kind of threat is this? How should the adaptive immune system respond? And it's been doing this for hundreds of millions of years, long before the evolutionary innovation of adaptive immunity came along. Insects rely on it entirely. Plants have their own version. Even single-celled organisms have rudimentary pathogen detection systems. The adaptive immune system, the one with the antibodies and the T cells and the exquisite specificity, that's the newcomer. It evolved only about 500 million years ago in the jawed vertebrates, layered on top of this ancient innate foundation. And that layering is important to understand. because it tells you something about how the whole system is architected. The innate immune system is fast but broad. It responds in minutes to hours, using pre-formed receptors that recognize general patterns. It can't adapt to a specific pathogen the way antibodies can, and it doesn't have memory in the classical sense. Or at least, that's what everyone thought until quite recently. But that's a story I want to come back to. The adaptive immune system is slow but precise. It takes days to generate a tailored response. But once it does, it's devastating in its specificity, and it remembers. The two systems aren't separate armies operating independently. They're deeply integrated, constantly communicating, each one shaping the other's behavior. The innate system activates and directs the adaptive system. The adaptive system, in turn, enhances innate effector mechanisms. Antibodies coating a bacterium make it far easier for phagocytes to engulf it, which is opsonization, the very process that resolved the Mechnikov-Ehrlich debate a century earlier. And that mention of innate immune memory, this is one of the most exciting developments in immunology in the last couple of decades, and it challenges something that was considered almost dogma. For the longest time, the textbook distinction was clean and simple. Innate immunity is fast but has no memory. Adaptive immunity is slow but remembers. Except researchers started noticing something strange. In certain organisms that have no adaptive immune system at all, plants, invertebrates, a prior infection seemed to provide some protection against a second encounter with the same pathogen. How? There are no antibodies, no memory T cells, no clonal selection. And even in mammals, epidemiological studies and vaccine trials started suggesting that certain vaccines, particularly the BCG vaccine against tuberculosis and the measles vaccine, seem to provide protection against unrelated infections, reduced overall childhood mortality beyond what you'd expect from just preventing the target disease. Something was happening in the innate immune system that looked an awful lot like memory. And that's exactly what it turned out to be, though not the kind of memory anyone expected. The person who really crystallized this idea is a Dutch immunologist named Mihai Natea, working at Radboud University in the Netherlands. Around 2011, Natea and his colleagues started publishing work that gave this phenomenon a name, trained immunity. And the name itself is interesting because it's deliberately provocative. Training implies learning. Learning implies memory. And memory was supposed to be the exclusive domain of the adaptive immune system, of lymphocytes, of clonal selection, of all that beautiful machinery we've been talking about. But Natea's group showed that innate immune cells, particularly monocytes and macrophages, could be reprogrammed by an initial encounter with a pathogen or a vaccine in ways that made them respond more vigorously to a second, completely unrelated challenge. Here's how it works, and it's genuinely elegant. When a monocyte encounters something like the BCG vaccine, which contains a live, weakened strain of Mycobacterium bovis, the cell doesn't just fight off the bacterium and go back to baseline. Something changes inside it. Specifically, the cell undergoes epigenetic reprogramming. Now, epigenetics. This is one of those terms that gets thrown around a lot, so let's be clear about what it means here. Your DNA contains all the instructions for making every protein your body could ever need. But not all of those instructions are active in every cell at every moment. Epigenetic modifications are like bookmarks and sticky notes on the DNA. Chemical tags, mainly methyl groups and acetyl groups attached to the DNA itself, or to the histone proteins that the DNA wraps around. These tags determine which genes are easy to read and which ones are tucked away, hard to access. They don't change the genetic code. They change how accessible it is. What NIAID's group found is that after exposure to BCG or to certain fungal components like beta-glucan, monocytes undergo shifts in these epigenetic marks. Genes involved in producing inflammatory cytokines, the signaling molecules that amplify immune responses, become more accessible. The cells are essentially primed. They haven't changed their receptors. They haven't undergone clonal expansion. They haven't generated anything like an antibody. But the next time they encounter a pathogen, any pathogen, they respond faster and more aggressively. They produce more tumor necrosis factor, more interleukin-6, more of the chemical signals that recruit other immune cells and ramp up inflammation. The cell has been trained. And here's what's really fascinating about this. It's not permanent in the same way adaptive memory is. Your memory B cells can persist for decades, maybe a lifetime. Trained immunity in monocytes seems to last months, maybe a year or two, because monocytes themselves are relatively short-lived cells. They circulate in the blood for a few days before migrating into tissues and differentiating into macrophages. So how does the training persist beyond the lifespan of an individual cell? The answer, it turns out, goes deeper, all the way back to the bone marrow. The epigenetic reprogramming doesn't just affect circulating monocytes. It affects their precursors, the hematopoietic stem and progenitor cells in the bone marrow that give rise to new monocytes. The training is passed upstream, so that newly minted monocytes emerging from the bone marrow already carry the enhanced inflammatory program. They're born ready. Think about what that means for a second. A vaccine you received, let's say BCG, given to you as an infant in many countries around the world, didn't just teach your adaptive immune system to recognize tuberculosis. It also reprogrammed the factory that produces your innate immune cells, giving every new monocyte that rolls off the assembly line a slightly heightened state of readiness. That's why those epidemiological studies kept showing that BCG-vaccinated children had lower mortality from all causes, not just tuberculosis. Their innate immune systems were running at a higher baseline, catching infections earlier, responding more forcefully, buying time for the adaptive system to spin up its precision response. Now, you might be thinking, if trained immunity makes your innate cells more aggressive, isn't there a downside? And yes, absolutely there is. This is the immune system after all, and by now you've probably noticed the pattern. Every powerful mechanism comes with a shadow side. If trained immunity can be beneficial, enhancing your ability to fight off infections, it can also be detrimental if it tips into excessive inflammation. There's growing evidence that trained immunity may contribute to chronic inflammatory diseases, atherosclerosis, for instance, where macrophages in arterial walls become hyperactivated and drive the formation of plaques. The same epigenetic reprogramming that helps you fight off a respiratory infection might, in a different context, contribute to the slow inflammatory damage that leads to a heart attack 20 years later. It's horror autotoxicus showing up again. That fundamental tension between protection and self-harm, just wearing a different coat. And this brings us to something worth pausing on as we think about where immunology stands right now in the 21st century. Because if you step back and look at everything we've talked about tonight, from Thucydides noticing that plague survivors could nurse the sick without falling ill again, to Jenner scraping cowpox into a boy's arm, to Pasteur's attenuated vaccines, to Mechnikov's phagocytes, to Ehrlich's side chains, to Burnett's clonal selection, to the discovery of T-cell subsets and thymic education, to Tregs and checkpoint inhibitors and pattern recognition receptors, and now trained immunity. What you see is a story of increasing complexity. Every time immunologists thought they had the picture, someone turned over another rock and found an entire ecosystem underneath. And the current frontier is maybe the most complex layer yet, because it involves the immune system's relationship with the trillions of microorganisms that live in and on your body, the microbiome. Your gut alone contains something like 38 trillion bacteria, roughly as many cells as make up your entire body. And these aren't invaders. They're residents. Many of them are essential. They help you digest food. They synthesize vitamins. They compete with pathogenic bacteria for resources and space. And your immune system knows they're there. It has to. It's in constant dialogue with them. This is where that concept of tolerance we talked about earlier becomes incredibly nuanced. Your gut immune system, and there's a huge amount of immune tissue in your gut, something called the gut-associated lymphoid tissue, or GALT, which contains more immune cells than any other organ in your body, has to perform an almost impossibly delicate balancing act. It needs to tolerate the commensal bacteria, the beneficial residents, while remaining vigilant against genuine pathogens that might arrive through the same route. And it needs to tolerate food antigens, the proteins in the meal you had for dinner, while still being capable of mounting a response against a toxin or an actual infection. Remember what we discussed about how the innate immune system's pattern recognition receptors control whether the adaptive system gets activated? This is where that principle becomes a matter of daily, moment-to-moment survival. The context in which an antigen is encountered, whether it arrives with danger signals or without them, whether it's presented by a dendritic cell that's been activated by pattern recognition receptors, or by one that's in a resting, tolerogenic state, determines whether you get immunity or tolerance. And in the gut, the default setting is tolerance. Dendritic cells in the gut tend to promote the generation of regulatory T cells rather than effector T cells, creating an environment of active suppression that keeps the peace with your microbial residents. When this system breaks down, the consequences are severe. Inflammatory bowel diseases like Crohn's disease and ulcerative colitis are, in essence, failures of intestinal immune tolerance, the immune system turning its weapons against the gut's own microbial community or against the intestinal tissue itself. And there's mounting evidence that disruptions to the microbiome early in life, through antibiotic use, through cesarean delivery, that bypasses the infant's exposure to the mother's vaginal and intestinal bacteria, through ultra-hygienic environments, may contribute to the rising rates of allergies, asthma, and autoimmune diseases in industrialized countries. This is sometimes called the hygiene hypothesis, though many immunologists now prefer the term old friends hypothesis, proposed by Graham Rook. The idea isn't that dirt is good for you in some vague way. It's that the human immune system evolved in the presence of a specific set of microbial companions, and that exposure to those organisms during critical developmental windows helps calibrate the immune system properly, helps set the balance between aggression and tolerance at the right point. Remove those microbial signals, and the calibration drifts. The immune system becomes more likely to overreact to harmless antigens like pollen or peanut proteins, or to turn against the body's own tissues. And this connects to one of the most exciting therapeutic frontiers in modern immunology: the deliberate manipulation of the immune system. We talked about checkpoint inhibitors for cancer, and those are a spectacular example. But the broader vision is even more ambitious. Researchers are working on ways to retune the immune system with extraordinary precision. CAR T-cell therapy, for instance, chimeric antigen receptor T-cell therapy, takes a patient's own T-cells out of their body, engineers them in a laboratory to express a synthetic receptor that recognizes a specific molecule on the surface of their cancer cells, and then infuses those engineered T-cells back into the patient. It's like giving the adaptive immune system a software update, programming it to attack a target it was previously ignoring. The first CAR T therapies were approved in 2017 for certain types of leukemia and lymphoma, and the results in some patients have been remarkable. Complete remissions in cases where every other treatment had failed. But the side effects can be severe, including cytokine release syndrome, a massive inflammatory reaction caused by the sudden activation of all those engineered T cells at once. The immune system, once again, demonstrating that its power is inseparable from its danger. On the other side of the coin, there's work on inducing tolerance, teaching the immune system to stop attacking things it shouldn't. For autoimmune diseases, for transplant rejection, for allergies. The dream is antigen-specific tolerance, turning off the immune response to one particular target without suppressing the entire system. Current immunosuppressive drugs, the ones used to prevent transplant rejection, work like a sledgehammer, dampening the whole immune system and leaving the patient vulnerable to infections and cancers. But what if you could selectively delete or suppress only the T cells and B cells that recognize the transplanted organ, while leaving the rest of the immune system intact? Researchers are exploring approaches using tolerogenic dendritic cells, engineered regulatory T cells, nanoparticles coated with specific antigens, all trying to exploit the immune system's own tolerance mechanisms with surgical precision. And here's where I think the story comes full circle, in a way that feels almost poetic. Because Jenner, back in 1796, was doing something remarkably similar in principle, even if he couldn't have known it. He was taking the immune system's own machinery, its ability to recognize, respond, and remember, and redirecting it. He didn't understand the mechanism. He didn't know about T cells or B cells or pattern recognition receptors or epigenetic reprogramming. He just knew that milkmaids who'd had cowpox didn't get smallpox. And from that single observation, that one thread of empirical knowledge, an entire science unfolded over the next 2 centuries. What strikes me most about the history of immunology is how often the right answer was staring people in the face long before they had the tools or the framework to see it. Metchnikoff watched a starfish larva engulf a thorn and intuited something fundamental about cellular defense decades before anyone understood how it connected to the rest of the immune system. Gruber and Perquet coined the word allergy and recognized that the immune system could cause disease, not just prevent it, years before anyone understood the mechanisms of autoimmunity. Gershon proposed suppressor T cells and was ridiculed, only for the concept to be vindicated decades later as regulatory T cells. Janeway predicted that the innate immune system must have pattern recognition receptors years before they were found. Over and over, someone saw a piece of the puzzle and tried to tell the world, and the world wasn't ready to listen. And maybe that's the most human part of this whole story. Not the molecules, not the receptors, not the elegant cascade of signaling pathways, though all of that is beautiful in its own way. The most human part is the stubbornness, the intuition, the willingness to be wrong, the occasional flash of insight that changes everything. Immunology wasn't built by abstract forces. It was built by people. Curious, flawed, brilliant, sometimes petty, often courageous people who spent their lives trying to understand how a bag of cells and proteins manages to tell the difference between self and not self, between friend and foe, between a harmless peanut protein and a deadly virus. And that system is working inside you right now. As you lie here in the quiet, your immune system is patrolling. Neutrophils are rolling along the walls of your blood vessels, sampling the environment. Macrophages in your lungs are sweeping up the dust and microbes you inhaled today. Dendritic cells in your skin and gut are monitoring the boundary between your body and the outside world, deciding what deserves a response and what can be safely ignored. In your lymph nodes, T cells are slowly circulating, each one checking the molecular dispatches brought in by those dendritic cells, looking for a match. Memory cells from every infection you've ever fought and every vaccine you've ever received are quietly persisting. Some of them in your bone marrow, some in your tissues, ready to reactivate in hours if they ever encounter their antigen again. Regulatory T cells are keeping the peace, suppressing responses that might otherwise turn against your own tissues. And in your bone marrow, right now, new immune cells are being born, differentiated, educated, and released into circulation, a continuous renewal that has been going on every moment of your life and will continue as long as you live. It's all happening without any conscious effort on your part. You don't have to think about it. You don't have to direct it. It just works. This vast, intricate self-organizing system that has been shaped by hundreds of millions of years of evolution and refined by every pathogen your ancestors ever encountered. It's not perfect. It makes mistakes. Sometimes it's too aggressive, sometimes not aggressive enough. But on the whole, on the vast majority of days, it gets it right. And that's why you're here. That's why any of us are here. So let that settle over you. All that complexity, all that history, all those discoveries and debates and breakthroughs, they're all describing something that's quietly taking care of you right now, in this moment, as your breathing slows and your thoughts begin to soften. You don't need to hold on to any of it. The details will be there whenever you want to come back to them. For now, just rest and let your immune system do what it's been doing beautifully, silently, for your entire life. It's got this, and so do you.